example of how I want to do it. And this slide will stay for the rest of the talk. So I kept having the same slide so that I can actually explain what I mean. So this is, let's say, the inside of your cell. You have the nucleus where there's your genes, your DNA. They get transcribed, they come out into the cytosol. On this, you actually translate the genes, just like translation of languages, you translate the genes into actual products. And these products then make proteins, and these proteins go to various places. And I've listed some of these intercellular bacteria. You might have heard of some of them, salmonella, because it's diarrhea. I'm sure some of you, after lunch, after dinner, I shouldn't say. And then there's some that cause tuberculosis, kills 10 million people a year. These are all horrible bacteria. So why is it that I think that they can become useful? Well, the reason is, look at the arrows. They all go to various places inside the cell. They all try to avoid the killing inside the cell. So when a bacteria comes, it normally goes to that place called lysosome, where it's very acidic. It's a place where you chop down the proteins and you kill the bacteria. The reason that these pathogens are successful is because they go to other places. They build their homes inside our cells, and they fight us because they go somewhere else. And what we are trying to figure out is how these pathogens do that. They're secreting some bacterial proteins inside the host cell that manipulate our host. So I want to put forward this idea that research and technology takes a long time to develop, right? What we are trying to do is to predict an epidemic. We are trying to predict a disease that's going to come and hit us and cause a lot of deaths. So can we do a little bit better than that? We can predict it, and we can find these tools that these other bacteria has already started manipulating, and then use that as a defense organism. So make sure that these pathogens cannot escape that route and can go straight to where it's supposed to go, which is the lysosome. So the idea would be, by using these as model organisms and model pathogens, can we then predict something that's going to come in the future? For example, when we heard about Ebola and we heard about Zika, do you think at that time, if the government gave the scientists money and said okay, make us the vaccine right now in 5 days, that's not going to happen. It takes a long time to develop medicines. So what we really try to do in our lab is to basically use these pathogens as tools to understand basic cell biology mechanisms that we can use for all pathogens, that we can actually give a broad covering and a shield for our cells, really. And one of the examples I put forward is that guy sitting next to me, Mike Bishop. He won the Nobel Prize. He works at UCSF. He used to work at UCSF. He just retired. His lab was right next to me. And he was working with that virus called Rouse sarcoma. And that sarcoma virus, that protein actually was the first oncogene that we actually discovered can cause cancer. So from a virus, you can learn so much that you can relate to other diseases. So I would like to thank, actually, the taxpayers' money, all of you guys that funds this kind of research. National Institute of Health is really your money, so thank you. And then also there are some private fundings and donor money that we receive to carry out these research programs.
Yay, another microbiologist. Do you work with all of those bugs, or which one is specifically?
I work with the one called Legionella pneumophila, this one. And the reason is that what I looked for is a toolbox, right? I wanted to find tools, and this one secretes 300 proteins inside the host cell, as opposed to these other that have between 5 and 40. So because it has the maximum, I chose that one as the model organism. And also, it's easy to manipulate. You can do genetics with it. So you want to use something that you can, play with, kind of.
Are there any examples of people modifying these different pathogens for therapeutic use?
Yeah, so they actually don't use the pathogen per se, but they use some of the proteins that these pathogens are. Because if you use the pathogen, it will cause the disease, right? So what they do is basically use the protein that is most relevant and use that as a therapeutic agent. So yes, that's been done.
Are there any examples we've heard of?
Yeah, so for example, there is a protein in a bacteria called cholera toxin. I don't know if you've heard of cholera. It causes again diarrhea, and cholera toxin is this toxin that the cholera secretes inside the host cell, and it blocks protein translation. So guess what happens when we have cancer? There's a lot of cell division. The cells proliferate and become a tumor, right? It becomes a big mass. And for that to happen, there's a lot of protein translation. So a lot of proteins get made very quickly. So people have used those toxins to direct it at those places to kill the translation. So that has been, a nice tool to start, use, right? So if you can take the thing out and can make it so that it is more directed, then you can make it useful. Also, the idea of antibiotics, right? So we know that, maybe some of you have heard that antibiotics kill bacteria. So when you have a bacterial infection, antibiotics should be good, right? But it's not as simple, because even take the example of cholera, right? Cholera toxin is inside the bacteria that carries it, right? E. coli sometimes carries cholera toxin also. And there's also Vibrio cholera, which is the original bacteria that carries it. But now let's say you had that bacteria inside your body, and the doctor gave you antibiotics. What will happen? The bacteria will die, it will release the cholera toxin more. So it will actually be worse. The toxin will actually come out more if you kill the bacteria. The other problem is that your gut microbiome, which is the bacteria that's inside your gut, is actually the helpful bacteria. That will also die when you use antibiotics. And those bacteria are actually very good in protecting you and are important in a lot of disease conditions to help you. So if you analyze all of those, then you have a problem. So bacteria, you should take antibiotics when it's absolutely necessary to fight the infection, but you have to take it with a lot of caution. Because also, the more the bacteria get used to the antibiotics, the more chances that they'll get resistant to it. Because think about it, the bacteria divides every 20 minutes. We as humans maybe divide once every 30 years. So we have a very bad disadvantage from just that, right? Because once they divide, they get their chance to change their genomes, right? And the change in their genomes makes them more resistant, right? So when you add antibiotics, they realize okay, this antibiotic is targeting this gene, I'll change it quickly so that it no longer is active. But we can't do that, right? We have to wait for generations for some little things to happen, so, yeah.
It's only sort of related, you mentioned cancer. I read a thing today in Consumer Reports that talked about laxatives to be used to keep yourself from getting cancer. Is there any connection to your, I guess, digestive system and moving stuff through there and cancer? I'm assuming it has something to do with the presence of all of these cells inside of you.
Well, I think a lot of people make a lot of generalized sort of statements.
It was about that long.
Yeah. It doesn't. It's not as simple, So cancer can be caused by various reasons, right? One of the biggest reason for cancer is genetic mutations. So you have the gene is a set of letters, let's say, right? And it's supposed to be translated in a certain way that is a useful protein, right? What happens is that when our cells divide, the gene has to be replicated properly so that you have the same sequence that was in the original mother cell, right? When this replication is happening, and it's happening very fast, it's happening all the time, right? And there is a proofreading process that reads that you changed it right, you did everything right? Let's say as we age, this proofreading process is not the same as robust as when we are young. So when we age, we get cancer. Some people get cancer is because their proofreading slows down, and so they're not able to proofread as much, and so the genes are changed, and the resultant protein is not a functional protein, and it causes some other activity, and that causes the cancer to happen. So by just having laxative is not, I feel like that's a very simplistic way of thinking, I would say.
It was a read the article kind of thing.
So you mentioned using these microorganisms as a tool. You didn't mention that you were changing the organism. Like, are you CRISPRing something, or is this?
No. So the reason I'm saying that I'm using the pathogen as tool is because I'm using the pathogen to study the proteins that are coming out of it. But actually what I'm using the tool are the proteins.
Okay.
So it's really fascinating. So I'll give you an example if you have like a little bit of time.
Yes.
So
Please.
let's say we found a protein in Legionella that was causing a weird modification to a host protein. By host I mean us, right? So it was causing this modification, and when I saw this, I was like, what is this modification? What is it doing to the host cell? So I looked for the domain that this bacteria had that was necessary to cause this modification. I searched the database. Guess what? Our own human cells have this protein that I first discovered in the bacteria, and it's called Huntington interacting protein E. So this protein came down, so Huntington's cause Huntington's disease, which is a neurological disorder. And people still don't understand how that disease is offset. Like, we know like it happens at their old age, but we don't know exactly what causes it. And now by studying this bacteria, which is not related to Huntington's at all, we've found a protein that is very similar to what this Huntington's protein act. And now we can look for modifications in Huntington's disease patients to see whether that's what I'm calling tools. So you can never know where your bacteria leads you to. It can go to cancer. It can, like Mike Bishop, when he was looking at the virus, he didn't think of cancer, but he found a protein that then became a really important player in cancer. So, but being a microbiologist is not just, at least the way I think of microbiology, is not just to think from the perspective of finding a drug. It's really to uncover new things inside the cell, because these bugs are super clever. They're doing that. I feel like they are more clever than us scientists, so we can learn from them, right? So I want them to be our teachers.
I just wanted, and Gully more questions, I wanted to thank you for explaining this in like super simple.
Yeah.
I was worried. I was worried.
Like,
I was worried that
usually when there's science talks, I'm like, and I probably get like 50%, and I feel I got like 85.
Okay. I'm sorry about the 15 then.
I think that's just not attainable.
You're talking about humans with your research, but how? But what I really want to know is, how long has this been going on? Do these kinds of things affect all living things? Is it something that's just part of every biological system?
Yeah, that's a great question. Actually, these bacteria have evolved much longer than we did, right? Like, so they're like, some of them are billions of years old, and they originally, the one I studied, Legionella, originally started off by being inside amoeba in just river pond water. It never wanted to actually kill humans, right? It never did, and then what we did.
That's just because humans didn't exist, so why would it want to kill
No,
them?
even when humans existed, it didn't want to do that. But then what happened was humans invented the air conditioner, right? And they put in the air conditioning main system, they had to put water reservoirs, and they took the water that was containing Legionella and they put it in the air conditioners. The air conditioners then spit out this air that is full of aerosols that contain Legionella.
This
and
is Legionnaire's disease.
That's Legionnaire's disease. And that was discovered in 1976 only. So it really didn't want to kill. I feel bad for it. I feel like the pathogens, they don't have any benefit of killing us. It's just that
But we insist.
we insist.
So I have a question. So you were talking about this Legionnaires, but also cholera, and I'm reminded of when UN workers went to Haiti and they kind of brought cholera to Haiti after the earthquake. So is there anything we can learn from that and from this other Legionnaires example about how not to just...
Yeah. So like the example of like if you have cholera and the doctor treats you with antibiotics, the cholera toxin comes out and then kills more.
Right.
So we, in our research, like not in our my lab, but other labs, have shown that if you just add magnesium, this ion, I'm sure some of you have heard calcium, magnesium, like tablets that you can like just take like over the counter. If you actually take magnesium, this cholera toxin cannot travel. So the cholera toxin has to get here for it to act. And when it comes in the plasma membrane, it can get stopped if you have enough magnesium inside the cell. So by just having an antibiotic treatment with magnesium, what it will do is it will kill the antibiotic will kill the bacteria, and the magnesium will block the cholera toxin. And that combination is a lot more effective than just, so we have identified mechanisms of how these work, and that helps us give this information to the doctors, who then will treat the patients.
I'm thinking also, like, are there unintended consequences? Like, is it unavoidable as far as just with what we do with our natural resources, with extraction and then just the movement of people?
No,
like,
I think it is avoidable if you boil the water that you drink and your sanitary health conditions are better, then yes, it's avoidable. But some of these cholera or salmonella outbreaks happen in third world countries where the sanitation and the healthcare is not as good. And so we have to also think from their perspective. We cannot, find a drug that's like, $10,000 per pill, and that's not going to have any effect. So yeah, but it's definitely avoidable. If you boil the water and you kill the bacteria, then it's fine. It will not cause anything.
Thank you so much.